Hi everyone, welcome to part 3 of our lecture series on the lymphatic and immune system. In this part we turn our attention to the adaptive immune system, which is quite extraordinary. In contrast to the innate immune system, the adaptive immune system is not necessarily present at birth. It is a system that can become more or less robust throughout your life, depending on kind of the exposure to various pathogens you will experience throughout your life. Also, it's different in that it's very specific. The cells of the adaptive immune system are able to pinpoint and target with an extraordinary degree of specificity, meaning cells might be able to target one strain of bacterium and not another very closely related strain of bacterium. Okay, so it can even differentiate between strains of bacteria or even very specific strains of virus. Some strains of bacteria might be pathogenic, others may not. It might be the same species of bacteria, but one might be pathogenic, one may not, and your body could potentially pinpoint the pathogenic strain and destroy it while leaving the other one intact. That's not something the innate immune system can do. The innate immune system kind of destroys general, pathogens expressing a general feature about them. Okay, also what's interesting is that this is an immune system that can detect, recognize, and destroy pathogens that don't exist today but might exist 10 or 15 or 20 years from now. And it's at this point you should think to yourself, how on earth does a cell with no eyes, no brains, no ears, can't see, how does it recognize a virus that doesn't even exist today but will in 10 years? How does it recognize a bacterium that doesn't exist today but might exist in a few years? It's pretty fascinating. We'll talk more about how it does this later when we talk about the B cell and the T cell receptor, which is coming up here in just a second. Okay, some of the important terms and concepts. The first is this word antigen. Antigen is a general word. There's a word used to describe generally anything that has a specific set of amino acids or a specific sequence of carbohydrates or nucleotides that can be recognized and bound. There are also on an antigen, a big particle that's recognizable. There are specific kind of micro parts of the antigen that are bound by lymphocytes in our body or other cells. These are called antigenic determinants. So there's a figure in your book which shows a protein. And that protein has many amino acids. It's a big long chain of amino acids that's folded into its tertiary or quaternary shape. But then above and beyond being able to bind specifically to the protein, there might be just a sequence here that's only a handful of amino acids long, 3, 4, 5, 6, 7, 8, 9 amino acids long, and that receptor might specifically bind just to that one region. So one antigen could be bound specifically by a variety of different receptors that are each binding to its own specific antigenic determinant. Another word you might hear when we're talking about this is this word epitope, which again just means the specific region on an antigen that is bound by a receptor. All right, the TCR and the BCR. These abbreviations stand for T cell receptor and B cell receptor. So there are a bunch of different types of lymphocytes which are called B cells. There are a bunch of different types of lymphocytes which are called T cells, and there's a variety of kinds of B cells and T cells. But all B cells will have a little B cell receptor on it, and all T cells will have a little T cell receptor on it. Now, what's interesting about this is that the gene that that codes for the production of the B cell receptor and the T cell receptor has a variable region on it, meaning it's a region on the gene that can be alternatively spliced in a variety of ways to make a whole bunch of different combinations of protein. So it might be just a handful of genes, but those genes could, through just random reorganization, make billions or billions and billions of different kinds of receptors. And that is exactly what our immune system is doing all the time. Right now in your body, you're making B cells that have billions of different kinds of B cell receptors. Each B cell will only have a B cell receptor that binds only one type of epitope or one type of antigenic determinant, but you'll have millions and millions and millions of different B cells that are made in your body, each with its very own specific binding pattern. And that goes for T cells as well. Now, this may seem on the surface a little wasteful because you might, in essence, make 1 million or 2 million or billion B cells that never actually bind anything because they each have their specific B cell receptor that binds something that's actually never going to be found in your body. That goes for the T cells as well. So it seems a little wasteful that you'd be making all of these cells, putting all this energy into making cells that never get used, never can bind anything. But because of the sheer random luck of the draw of how that process occurs, it enables us to have somewhat of a ability to recognize and destroy pathogens that exist in the future that don't exist today. And so while we can't predict, our immune system can't predict what viruses or bacteria will enter the scene and act accordingly, just because there's such a random assortment of B and T cells in your body, it's likely that when that pathogen comes on the scene 10 or 15 or 20 years from now, you might have a cell with a receptor that can recognize it, just because of the sheer random luck of the draw. So it's really a pretty beautiful system in that sense, and quite extraordinary. Okay, there is another molecule we should talk about here called the major histocompatibility complex, or MHC. What MHC is it's kind of like a flagpole that puts antigens on the surface of a cell. There are two kinds of MHC. There's the MHC molecule 1 and MHC molecule 2. They both have kind of a transmembrane domain that anchors it into the plasma membrane. And then they have kind of this little region here where there's a cleft that can bind peptides, or little sequences of proteins. So in a way, any antigen that well, maybe I'll back up. Antigens that have that are endogenous antigens, maybe I should put that word up here because you might see this as you're traversing the veil of immunology. Endogenous antigens are ones that have actually been absorbed and kind of infected a cell. And usually, like if a virus or a bacterium invades a cell and the cell is infected with it, those would now be endogenous antigens, and they usually get presented on MHC class 1. So if there's a little particle on MHC class 1, it tells the body, ooh, that cell is infected. And I'll also say that fragments of all parts of the cell are usually displayed on MHC class 1. And so when a virus or a bacterium goes into the cell, without exception, it also gets presented. So there will be most of the antigens on the surface of a normal cell will be antigens that aren't from a pathogen, but if a pathogen does invade, it also too gets expressed on the surface through MHC class 1. MHC class 2 then is used to present exogenous antigens. So these are antigens that didn't necessarily infect or invade the bacteria or the cell, the immune cell, but perhaps the immune cell was able to phagocytize it in a controlled manner, and without becoming infected, it took those bits and processed them a little bit differently and then presented them on an MHC class 2. Usually, like if a let's say a virus or a bacterium gets phagocytized, rather than it kind of wreaking havoc in the cell, there's a controlled way to wrap it around with a bit of plasma membrane to create a vesicle that is kind of professionally then broken down without affecting other parts of the cell. And when this kind of endocytosis occurs, the cell is able to recognize it and display it not on MHC class 1, but instead on MHC class 2. So in summary, MHC class 2 would be on a cell and present antigens which have been kind of professionally phagocytized. MHC class 1 is going to be a molecule that presents antigen of anything found in the cell. And if a pathogen gets into the cell, it also will at some point be presented on the outside of the surface of the cell with all of the other bits and pieces that are normally found in the cell. Okay, this will hopefully make sense as we get further into our discussion here. But one thing I like to point out is this famous experiment. You can't quite get through a lecture in immunology without bringing up this experiment. This was done in 1995 by Klaus Wedekind was the leading investigator here on this subject. This is sometimes referred to as the sweaty t-shirt experiment. And this isn't going to be on the exam or anything, so if you don't really care to learn about this, you can go ahead and skip ahead. But I like to talk about it because it's a super interesting experiment. What happened is that this researcher asked a couple of dudes to wear t-shirts and wear them for 2 days straight, not wearing any deodorant, any cologne or scented soap or anything. And then at the end of the 2 days, he put all of these t-shirts in a box, and he put them in the same, all in the same box, and asked women to come up and smell the sweaty t-shirts and write down which t-shirt they thought they were most sexually attracted to. And what was really interesting is that with a certain degree of statistical significance, women were generally most attracted to t-shirts from men that had a MHC complex that was most dissimilar to their own. So even though there's only two classes, MHC class 1 and MHC class 2, there are some variabilities in the MHC molecule itself. And for whatever reason, women were attracted to sweaty t-shirts of men with an MHC different than their own, most dissimilar to their own. This has also been repeated in 2005, and similar results were found. So kind of goes down as a famous experiment just because of how interesting the experiment is. Okay, one last thing is this whole concept called antigen presentation. We already talked about how antigens are presented on MHC. And antigen presentation refers to the ability of a cell to take in an antigen, break it down and sort it out and put it on the surface of its cell on the molecule MHC. Now there's a difference between what's called an antigen presenting cell and a professional antigen presenting cell. An antigen presenting cell is a cell that just presents antigen on it. It's not really clear if it's going to be MHC class 1 that's holding the antigen up, or if it's going to be MHC class 2. But generally, antigen presenting cells could be any kind of cell that presents antigen. But when we say it's a professional antigen presenting cell, then we're clarifying and we're saying that this is a cell that was able to take in the antigen, professionally break it down, and present it on MHC class 2 specifically. Okay, so keep that in mind. Because that will be kind of a different signal. There's a big difference between a cell that is presenting its antigen on MHC class 1 and a cell that's presenting its antigen on MHC class 2. The latter, MHC class 2, is done by what we call a professional antigen presenting cell. There are here's a good picture to show kind of the difference between these two pathways. This is a picture showing a dendritic cell, which is a really, really important antigen-presenting cell in the adaptive immune system. And an antigen-presenting cell, such as a dendritic cell, is pretty impressive because it can do both pathways. This is showing a pathogen that has invaded the cell, and with an antigen-presenting cell such as this, it doesn't necessarily have to be an invasion. It could be professionally absorbed through endocytosis. But in either case, a cell like that could be presented on MHC class 1. A cell such as this one could also absorb the pathogen in a vesicle and then add fuse with a lysosome containing vesicle to break it down, and then ones that have been broken down in a vesicle like this do not get loaded up onto MHC class 1, but rather they get loaded up onto MHC class 2, which I guess you can't really tell the difference between these. Oh yeah, this would be MHC class 2 here. So this is kind of interesting. Normally, MHC class 1 will be on showing pathogenic antigens of infected, cells that have been infected with a virus or a bacterium. In this case, though, if we're dealing with a dendritic cell, dendritic cells are really good at what's called cross-presenting, which means that they can professionally absorb an exogenous pathogen or an endogenous pathogen. They can so if this was just like viral particles outside and they went in, it can actually display them on MHC class 1 and MHC class 2. So normally when we see a antigen from a pathogen displayed on MHC class 1, it's kind of a signal that it's a cell that's been infected. But in this case, when it's a dendritic cell that is cross-presenting, it can also present its antigen on MHC class 1, but not actually be infected. It has total control of the situation. So that will kind of clarify things later as we get more into this mechanism. So I do like to point that out. What can then happen is that this dendritic cell can, these dendritic cells are usually found in the periphery of the body, like they're found in the stratum spinosum of the epidermis, they're found in the lymph nodes and, or just, in Peyer's patches, those by the digestive tract, so they're found all over the body. If it does absorb a pathogen, it usually goes into the lymph nodes where it can show a whole bunch of lymphocytes what is found. Then what happens is that the T cell receptor of a T cell or the B cell receptor of a B cell, but usually the T cell receptor of a T cell can attach to the same antigen if it has a receptor that binds specifically to that antigen. There's also a co-receptor next to the T cell receptor which binds to the MHC to help stabilize this whole bond. We'll talk about that later. Okay, so here's kind of the two pathways. There's antigen that's acquired from the inside of a dendritic cell. There's antigen acquired from outside of the dendritic cell. And then in either case, both are presented on the outside of the cell. Okay, here's a nice summary showing the different types of antigen presenting cells. Those that have class 1 MHC are many. Pretty much most all of the cells in your body will display antigens on class 1. And the purpose of this is really to help the body know what kind of cells each cell is. So if there's a liver cell, it will present a whole array of antigens that are specific to the liver. If it's a pancreatic cell, it might present a whole bunch of antigens that are specific to the pancreatic cell. So it helps the body kind of differentiate between cell types. And then just keep in mind, if something invades and infects the cell, it too also gets presented on class 1. These will usually stimulate what's called cytotoxic T cell response. We'll talk more about that later, but basically it's just a certain type of T cell that's really good at destroying cells that have been infected. Okay, class 2 is then going to be on B cells, dendritic cells, and macrophages. So these are more what we would call professional antigen presenting cells, because they can take up a pathogen and not display it on class 1, but instead display it on class 2. These are usually phagocytic cells, and it's through that process of phagocytosis that these pathogens are able to be professionally digested and then presented on MHC class 2. They have a little bit of different roles. The dendritic cells are really good at bringing antigens to lymph nodes. Okay, so they're the kind of the champions of that. And then 2, dendritic cells, when they get to the lymph nodes, as we talked about in our earlier chapter, they can actually anchor themselves in to the lymph node and let just cells brush by all day long as that lymph flows through the lymph node. And like a spider in its spider web, it can kind of catch any particular T cell that might bind to the pathogen that it's found. Okay, so that makes it really good at showing its antigen to lots of different B and T cells to help start an immune response. Okay, this can also stimulate presentation at the primary infection site. So macrophages are less well suited to travel into the lymph and get into lymph nodes. But if there is a regional infection, like on the outside of your skin where you just had a cut or something, there's likely macrophages there that can present antigen that they found in that local region. The B cells, we'll talk about these. These are the kind of the champions of making antibodies. So we'll talk more about that in a minute. All right, a little note here about how T cells are made. It's actually pretty extraordinary, and it's quite important to understand how they develop so we can understand how they function. When a T cell is made, it starts off as a thymocyte. So this is kind of the progenitor to a T lymphocyte. So we call it a thymocyte. These are what are made in the bone marrow, same with all of the other white blood cells. After it's made, though, in the bone marrow, a thymocyte will travel to the cortex of the thymus. The thymus is the gland that kind of sits on top of your heart. And in the cortex of the thymus, it will go through what's called positive selection. How this works is that there are a whole bunch of thymic epithelial cells inside the thymus, and the thymocyte, this thing here, will go around and it will bump into probably hundreds and hundreds and hundreds of thymic epithelial cells. Thymic epithelial cells have MHC class 1, and they display all of their, all of the antigens, which would be pretty normal for a regular cell in your body. And if there is a thymocyte that is not able to bind to the MHC, it's destroyed. And it likely means that it doesn't bind because it's got a faulty T cell receptor. So we want to get rid of any of these thymocytes that don't really have a good T cell receptor. So only those thymocytes that have a working receptor that can attach to MHC are positively selected, and then they will travel to, they'll stay in the cortex of the thymus, but at this point they will develop a CD8 receptor and a CD4 receptor. CD8 and CD4 are called co-receptors because they are not the T cell receptor, which is what we're seeing here. They're kind of this receptor that's right on the side of the T cell receptor that can help to stabilize a bond between the T cell receptor and a potential antigen that is presented by an antigen presenting cell. CD8 bearing cells are usually the ones that become cytotoxic lymphocytes. CD4 cells are usually the ones that become helper T cells. Okay, but in either case, after then going through the positive selection process, we basically end up with a thymocyte that has both CD4 and CD8. So it hasn't differentiated yet into a cytotoxic T lymphocyte or a helper T lymphocyte. From here, the lymphocyte will travel into the inner portions of the thymus. And here's where it goes through a very important process known as negative selection. And it's going to kind of go through the same rigmarole. It's going to go through and brush against a whole bunch of cells that have self-antigen presented on the surface. And so this is done there's dendritic cells in the thymus, and there'll be hundreds and hundreds of dendritic cells, and the thymocyte will brush against all of these dendritic cells and become well acquainted with all of the antigen that's presented on the surface of a dendritic cell. The dendritic cell in this case is not bearing foreign pathogenic antigens. It's just bearing self-antigen, the antigens that are supposed to be in the body. They're antigens from all of the different, it's a sample of antigens from all the different body cells. Now here, if either CD4 or CD8 binds to the MHC, or the T cell receptor binds to the antigen, it actually will destroy those cells. It's very important by the end of this whole process that the only T cells that are allowed to circulate around in the body are T cells that bind non-self antigens. T cells are really good at destroying whatever they bind to after they've completed their, their boot camp in the thymus. And so if there are T cells running around in the body binding to stuff that's supposed to be in your body, it will target it for destruction. So this is a very important step. It eliminates any T cell that would bind something that's supposed to be in your body, so that it ensures that the T cells that do finally get out of the thymus only bind stuff that's not supposed to be in your body and target those things for destruction. So you can imagine that a whole bunch of T cells get destroyed here. Many, 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 many T cells get destroyed because there's a lot of receptors that can bind self-antigen. So we're losing a lot of T cells here, but they get recycled, and the steady stream of new thymocyte recruits continues coming into the thymus. On the other hand, if we do have a T cell which does not bind any of the antigen, then it graduates from boot camp and it is able to leave. And at this point, it's still a thymocyte. It still has CD4 and it still has CD8. So in reality, we should probably draw a little CD4 and a CD8 on here. But it can be certain that the T cell receptors, or hopefully, if everything's working okay, it'll be certain that the T cell receptors don't bind anything that's supposed to be in the body. These cells will then be allowed to leave. They will, at this point, differentiate into either a CD4 thymocyte or a CD8 thymocyte. The ones that have CD4 on them become what we call mature T helper cells, but we still would call these, even though they're mature, we call them naive. And naive means that they haven't actually encountered an antigen yet. So there's T helper cells, and then there's the cytotoxic T cells. These are the ones that have CD8, and these are also naive because they haven't encountered a pathogenic antigen yet. These will then be allowed to leave. They'll go travel into lots of different places, but a real great place is the lymph nodes. So you're kind of seeing this here, but they'll travel around to the different lymphoid organs, the spleen or the mucosa-associated lymphoid tissues or the lymph nodes, where they will be ready for battle. There's what's called a thymic blood barrier, which ensures that the T cells that are inside the thymus are only able to leave once they get to the very innermost portion of the thymus. There's blood vessels that they can escape out of and go into the body. And all of the T cells that haven't undergone this training are trapped in the thymus. They're not able to get out, which is good because you don't want T cells running around your body that haven't gone through this extensive training. Okay, there are a variety of different T cells out there. There's a cytotoxic T cell. Its main target is infected cells of the body, and it is cytotoxic, meaning it literally just destroys a cell. And it destroys cells that are usually bearing pathogens that are intracellular. So this is a cell that's been infected with a virus or infected with a bacterium. The T cell has on its surface a CD8 molecule, and the CD8 molecule will bind, is a co-receptor that binds to class 1 MHC on the antigen presenting cell, whether it be just the regular old cell in the body or perhaps even a dendritic cell that's cross-presenting. It will release perforins and granzymes. We talked about these earlier. This is very similar to the NK cells, and I still am hazy about what the real difference is between a cytotoxic T cell and an NK cell. So if you find that information out, just let me know. It's also difficult because this immunology is a place where the information is always changing. So keep in mind the things we're learning today could be very well different in a couple of years from now. Okay, perforins poke holes in the membrane, and then granzymes can get into the cell and then start destroying proteins. There's also this thing called the Fas ligand. There's T helper cells, which are called T helper cells 1, and they are going to be binding to macrophages or professional antigen presenting cells. And they help to strengthen the macrophages. They help to strengthen the immune response. We'll see, I've got a diagram here coming up that just shows how wide-reaching the effects of T helper cells are. They usually are going to target extracellular pathogens, ones that have been professionally absorbed. So these would be virus or bacteria that are haven't infected a cell but are in the fluids and have been eaten by a macrophage. Generally, these professional antigen presenting cells will present their tasty meals that they have eaten on CD4 molecules, and CD4 is bound by class 2 to stabilize that interaction. And they'll release these things, which I'm not going to ask you to remember all the different cytokines that are released, but depending on which cytokine is released, it can have a different effect, which I'll show you in a minute, a figure that I've found. Okay, T helper 2 cells, they like to bind B cells, and they also help B cells. They're going to help B cells, and then when they activate B cells together, the main effect is to seek out and destroy anything that's outside of the cells. So this would be, again, parasites or virus or bacteria that have not invaded and infected a cell yet, but they're still in the fluids of the body. The B cells will express CD4, same as the macrophages, and that CD4 binds class 2. And when a Th2 cell binds to a B cell, it activates it by releasing all of these cytokines, which, again, I'm not going to ask you to memorize these. Okay, there's also regulatory cells, which are more recently discovered, and not a whole lot is known about them, but they do bind to T helper cells, and they're actually going to help to suppress the immune response. So after an immune response has begun, it's kind of important to then start to slow it down so that it doesn't become a very robust, over the top immune response. So these kind of regulate the immune response to make sure it's not too strong of a response, that you're not destroying too much at too quick of a time. You want to be able to just destroy the pathogen and then kind of halt the response after that. So these cells play a role in regulating that. They've got CD4 and CD25 on them, and again, not really sure what they bind to, but they do release, when a T regulatory cell attaches to a T helper cell, it releases these cytokines to help slow down the immune response, which, again, you don't have to know. This is a really great table, so if you get lost in this whole discussion of the adaptive immune response, please come back and find this table. One side of the adaptive immune system is called cell-mediated immunity, and this refers to the tactics that are used to destroy cells that have been infected. Now, this is a pretty complex process, and it's actually kind of hard for me to get my mind around it because a lot of the things that I'm reading don't quite line up. But I'll try and put together a story here that, for the most part, reflects the current research out there. All right, so this picture in your book is showing an activated T cell destroying an infected cell, and that's generally what happens, is that once a T cell is activated, it can go around and it can destroy infected cells. It does this by, first, the T cell receptor binding to the antigen that's being displayed by the infected cell. And then also the CD8 co-receptor generally binds to MHC class 1 to stabilize this bridge. And then what happens is that once these binding events happen, the T cell will release cytokines that destroy the cell. But what's less understood is the process by which T cells are activated. Before a T cell is activated, in this case a cytotoxic T cell, it's called naive, meaning it hasn't come in contact with an antigen yet. For a naive T cell to become activated, there's a couple different models out there, and I'll explain kind of the two signal model that I've been reading about. And basically, the two signal model says that in order for a naive T cell to be activated, it has to first come in contact with the antigen, but then in addition to that, so there's a first signal in which the T cell receptor binds to the antigen, and then likely also the CD8 would bind to MHC class 1, which is presenting the antigen. But then there's some research that suggests the second signal is also required for a stronger response here by the cytotoxic T lymphocytes. And that second signal involves a molecule called CD28 and then binding to these molecule is called CD80 or CD86. These, as far as I understand it, are on professional antigen-presenting cells, namely the dendritic cells. And so what this means is that usually for a naive cytotoxic T cell to become activated, in order for it to be a strong response, it needs to be first presented the antigen by a dendritic cell, not just a regular infected cell. So the dendritic cell would first present to the naive T cell. This is because the dendritic cell is the one that can create this second signal because of the fact that it bears these CD80 and CD86 molecules. And so the dendritic cell will bind to the naive T cell. It will cross-present its antigen on MHC class 1. Remember, MHC class 1 is normally just on an infected cell, but in this case the dendritic cell has professionally absorbed it and is kind of mimicking like it's been infected, but it's truly not. That's called cross-presentation. The dendritic cell brings it to the naive T cell, and so then the first signal occurs and then the second signal occurs. At that point, the naive T cells become activated, and it's at this point it's not quite clear to me. You would expect that the T cell, once it's bound to the dendritic cell, should actually destroy the dendritic cell because it treats it like it's a cell infected because of the fact that it's bearing CD8. But I don't know if entirely that is the case. If it is the case, then the dendritic cell has lived a good life because it's activated a cytotoxic T cell. But I think actually there's a delay in this process that allows the dendritic cell to escape, but it's not quite clear to me. In either case, after the dendritic cell turns the naive T cell on, it will then be active, but there's a couple other things that happen. One is called clonal selection and expansion, which just basically means that because this T cell with this specific T cell receptor is the one that binds the new pathogen, it goes through a whole bunch of divisions. At this point, the naive cell is maybe 1 out of a billion T cells, but because that 1 out of a billion is the one that binds the new pathogen invading the body, we need to make many copies of it so that it can fight the good fight. Okay, one other quick note is that instead of that second signal, the CD28 binding to a CD80 or 86 molecule on a dendritic cell, this step can be replaced by cytokines that are released by a T helper cell. So here we kind of are carving out 3 ways that a naive cell could become activated. One, either just by binding an infected cell, although I get the sense that's not a very strong activation signal, and so maybe the immune response isn't as strong. It could be activated by a dendritic cell that's cross-presenting, and in that case that's a little bit of a stronger immune response. And if not the dendritic cell, it could also be activated by a T helper cell that's releasing the appropriate amount of cytokines, the appropriate kind of cytokines that would activate the naive cytotoxic T cell. Okay, so assuming we've made it through this activation process, what an activated T cell does then is a little bit more simple. Now that it's become activated, it just goes around and binds any cell that is bearing the antigen of the pathogen that it's infected it, and then the T cell, if the T cell receptor binds and the CD8 binds the MHC, it targets it for destruction by either releasing perforin or granzyme or binding to the FAS molecule with the FAS ligand or ligand. Right, so that's a little bit about how the cytotoxic T cell works. There also needs to be a way to destroy virus and bacteria that are not inside of a cell, but instead in the fluids of the body. We need to be able to get rid of both. This usually, when we talk humoral immunity, we are referring to the secretion of antibodies, because antibodies are really good at getting into the fluid of spaces of the body, the small nooks and crannies of the body, to seek and destroy very specific pathogens. The way that this works is that still you have an antigen-presenting cell. Here we're seeing a dendritic cell. That dendritic cell could also be presenting MHC in class 2. what MHC class 2 will do is it's important because that's what's bound by T helper cells. And again, remember, you'll have a naive T cell which comes in contact with the antigen, and then through a variety of processes will become activated, and then it can bind to an antigen presenting cell. And what happens is that the T cell receptor will bind to the antigen just the same as it did with the cytotoxic T cell that we were just looking at. But here, keep in mind that a T helper cell, not a cytotoxic T cell, but a T helper cell also has a co-receptor, but it's not CD8, it's CD4. So the big thing to remember in your notes is that CD4 binds MHC class 2, whereas CD8 binds MHC class 1. Now, if we have these two events occurring, the T cell receptor binding to the antigen, the co-receptor binding to the MHC, then usually this will cause cytokines to be released to turn this cell on so that it can go do a whole bunch of stuff. And here is where it gets a little complex. Again, this is a hard chapter to talk about in an A&P class because things that involve molecular biology are always changing the more that we find out. That's different than other organ systems of the body, where we pretty much know how the heart works and how the lungs work on a macroscopic level. But here everything's changing. So let me just show you how complex some of these could be. Here, this is showing the same interaction between a dendritic cell and a T helper cell. But here they're not just showing us the T cell receptor and the MHC and the co-receptor. There's also CD8 and CD80 that bind. There's OX40 and OX40L. There's CD28 and CD86 that could bind. There's these molecules that could bind. So it's in reality a far more complex, and even in the years that have gone since I took a class in immunology in graduate school, the landscape of these interactions looks totally different. So it's a little hard to talk meaningfully about this chapter in this class because I know it's likely going to be different probably in a few years. And the more we find out, the less easy it is to navigate how these immune responses work because they're so interconnected and complex. But I do want to just kind of present kind of a scheme here that I thought was kind of interesting. This is from a textbook that was published in 2019, so it's a little bit more recent. And they are saying that when there's a T cell that's presented antigen, there's three signals that happen. The first is that the T cell receptor binds to the peptide that's expressed on MHC. They also kind of describe that the importance of the co-receptor is a little bit more minimal. Here there would also be a co-receptor that would bind to MHC, but rather than that being integral to stimulating this response, it really just helps to stabilize this interaction. They also say, so they talk about that being signal number 1, and signal number 1 is the first thing that needs to happen, but the second thing that happens is signal number 2, which occurs between this molecule known as CD28 and either CD80 or CD86. These are additional molecules present on an antigen-presenting cell. This second signal, after the second signal, leads to clonal expansion. That's going to mean that this T cell starts dividing. And then there's going to be a third signal. So depending on other molecules that bind to the antigen-presenting cell, there could be a third signal and a whole bunch of different cytokines which are going to cause this cell to become what it needs to become. So the T helper cell, or the T cell, could become either a T helper 1 cell, or it could become a T helper 2 cell, or the T cell could become a cytotoxic T cell. It kind of varies depending on what cytokines are released here. And then after all of those cytokines are released and there's the last differentiation of the cell, then these cells will generally go to the periphery of the body. They'll go to the place where the infection is occurring. Th1 cells are supposed to be helpful against virus and bacterium that are intracellular. So in that sense, they're kind of similar to a cytotoxic T cell. T helper cells, too, are good against extracellular pathogens. So this would be like parasites or bacteria or toxins or allergies, things that haven't quite been into the cell yet. So that's just to give you an idea of how really complex this starts to get. But for now, we'll just leave it here that when this event happens, a T helper cell is bound to MHC class 2. It releases cytokines that will cause further differentiation of the T cell. Here's another figure that I was able to find that came out of this paper, showing just how many different lineages could come out of an activated T cell. So again, you have the naive T cell becoming an activated T cell, and then depending on the cytokines that are released in this interaction, or rather this whole interaction, here are the different lineages of T helper cells that could be made. You could have the Th1 cells that release these cytokines right here. You could have the Th2 cells that release these cytokines. You could have Th17 cells that release these cytokines. You could have T regulatory cells that release these cytokines, and you could have TFH cells that release these cytokines. And they have all different types of roles. You know, the Th1 are involved more in cell mediated immunity, which is what we were just talking about earlier, the intracellular, destroying the intracellular pathogens. The T helper 2 cells are really good for humoral immunity, activating B cells, which we'll talk about in a second. And then here are our T regulatory cells. And then you also have these TFH cells, which I've never heard of before, that are also going to be involved in B cell activation. So this does get quite complex. I'm not going to ask that you know all of these different lineages. You can, for your knowledge, just keep it to what's presented in the textbook. In the textbook, they really just describe T helper 1 cells and T helper 2 cells. They describe T regulatory cells, and they describe T cytotoxic T cells. So knowing what each of these cells does, as presented in the textbook, will be what I expect for you on this class. But I do like to just show you this, how complex it gets. All right, so one very important part of the humoral immunity is activating a B cell. B cells are the champions that make antibodies, and B cells can either be activated by T cells through a process known as T cell dependent B cell activation, or B cells can be activated without T cells in a process known as T cell independent B cell activation. We'll talk about both of these here briefly. In B cell activation, it looks something like this. You have a B cell. Sometimes it's important that the B cell has already encountered an antigen. That's called B cell sensitization. And usually this happens, antigens that are T cell dependent antigens are ones that are more specific. They're not just heavily repeated. So it would be like a smaller antigen here. But in either case, this is called sensitization if the B cell has already come in contact with an antigen, and that primes it to then receive a helper T cell, which has also come in contact with that. So, or sorry, let me back up. Because the B cell has come in contact with the antigen, it will present that on MHC class 2. Remember that B cells are phagocytic, and so they can actually take in and phagocytize a pathogen and present that on MHC class 2. That's what would be the case after sensitization. And then if that's the case, then a helper T can then come up, and a helper T is one that has already been activated as well, because it has to, remember, go from being a naive T helper cell to an activated T helper cell. And so if you have two things, the B cell which has seen the antigen, and a T cell which has been activated because it too has also been presented the antigen, then the T helper cell, which is now active, can bind onto the antigen presented by the B cell, and then that will lead to the clonal expansion and proliferation of whatever B cell this is. Now this is important because the B cell receptor, this receptor on the surface of a B cell, is actually an antibody. It's either an IgD or an IgM class antibody. We'll talk about the different classes here in just a minute. And so then what happens is that there'll be what are called memory B cells, those that stick around to help strengthen a secondary immune response. But then also what happens is that the B cell will differentiate into these, which are called plasma cells. And a plasma cell will actually detach its B cell receptor, and now you have free-floating antibodies in the serum. The free-floating antibodies are those antibodies that will bind to whatever this antigen was that was first, bound by the receptor. So that's why antibodies will specifically seek out and destroy a pathogen, because remember, out of the billions and billions of B cell receptors on billions and billions of B cells, one specific B cell receptor bound to the pathogen, and then this cell got activated so that that specific B cell receptor is now secreted as an antibody, and that will still have the same affinity for the pathogen that it originally bound. So these go out into the body and they destroy pathogens. T cell independent B cell activation can also occur, and usually what happens in this case is that the B cell will bind an antigen. The key here is, though, the B cells that are activated without T cells have to bind to a specific kind of antigen. And specifically, it's called an antigen that has repeating epitopes. So meaning there are many of the same epitope found on the bacteria. Usually these are found on the cell wall. And so if we have a specific antigen like this, which is called a T cell independent antigen, meaning there's a lot of repeating epitopes, then in that specific case the B cell can activate itself and turn into a secreting cell. In this case, it secretes a certain kind of antibody called IgM. Again, we'll talk about these different classes. But the importance is that multiple B cell receptors are attaching at the same time because of the fact that there are these repeating antigens. If it's just a very specific antigen only found on kind of one part of the cell, and it only binds to one B cell receptor, then it has to be go through the rigmarole here of the co-stimulation with a helper T cell. Okay, now this process is usually pretty regulated, especially this process where the B cell has to first bind an antigen, become sensitized, present that antigen to a T helper cell. The two come together that release cytokines that turn this on. Well, this is important because this is a very strong response. A plasma cell can release thousands of antibodies every second. And so if you imagine you have clonal expansion, you have many clones of the original B cell that was activated, and they're each secreting thousands of antibodies every second, that becomes, something to the tune of millions of antibodies per hour released into the serum, and those travel everywhere into the body. So we definitely don't want antibodies to be made that will bind to something that is in our body, because antibodies generally target things for destruction. So that's why it requires so many steps. It's kind of a fail-safe mechanism so that we don't accidentally turn on a plasma cell that secretes antibodies that can bind to our own body's structures. Okay, a little bit more here about antibodies. What an antibody looks like is this. They have what's called an FC region on them. I forget what this stands for. I think it's like fractured crystallography or something region. But this is the region that's attached to the surface of the cell. And then on the outer region, they have what's called the variable region. Now this is the region, it's called variable because this is the gene that makes this protein is a gene that gets rearranged a lot. And so specifically the proteins that are made here in the variable region are made from the shifting portions of the gene that are continually rearranged so that it makes a whole variety of proteins here at the variable region of the immunoglobulin or the antibody. So these can bind a variety of different things, and each antibody has two binding sites that will bind the same antigen. Here are the different classes of antibodies that can be formed. The first is IgM, and usually this is the first type of antibody that is that is released if a plasma cell is activated. These are really good at destroying stuff because they have 10 binding sites. There's 5 antibodies all attached to one another at their FC regions, and that's going to lead to 10 binding sites. So this can just go out and just bind pathogens like crazy. So that's kind of one of the reasons why it's the first one that's bound. It's a bigger antibody, so it's a little bit more clunky. It's harder for it to get into smaller spaces, but initially when there's just a lot of free roaming pathogens in the fluids of the body, it does a great job at going through and destroying those all. It's also going to fix complement, and that is going to lead to the first way that, not necessarily the first, but it's one of the ways that antibodies work, is that they are the thing that start that whole complement cascade. Remember, complement, the ultimate result is to destroy cells. Okay, usually what happens then is that on a secondary immune response, less of the IgM is made, more of this one here called IgG is made. IgG is a very simple class of antibody, just having only 2 binding sites. It's very small, so it can get anywhere in the body. This one is the one that can actually cross placenta because it is so small. This one also fixes complement. This one here is interesting because it's got this available FC region that can bind to an FC receptor on something like a phagocyte. So in this scenario, let's imagine we have a phagocyte of some kind, and there's the FC receptor on that phagocyte, and here's our little bacterial cell, and antibodies have bound to it. Well, that FC receptor can go, oh, I see an antibody, and it can bind to the FC region of the antibody, and that brings that bacterial cell nice and close to the phagocyte so that it can be destroyed. So here we kind of see another way that antibodies work. They work through opsonization, making things more readily phagocytized, making them easier to be, creating handles, if you will, for a phagocyte to grab onto so that it can destroy it. We also see here that these play an important role in neutralizing toxins, and this is going to be another way that antibodies work. And to neutralize a toxin is just, it means that the antibodies will surround the toxin or surround the virus or surround whatever it is so that it can't bind to anything. So there's a picture in your book here that shows something that's really cool that happens. This is showing the small intestine, and it's showing the Peyer's patches. Peyer's patches are places where there's B cells and T cells underneath the surface. An interesting that happens here is that antigens are able to kind of get in through these passageways, which are called M cells, in the mucosal lining. So normally we have, pretty tightly bound epithelial cells in something like the lining of the small intestine, but every once in a while there's like these little trap doors, places where antigens can come in. But it's kind of funny because right in these regions where the pathogens are allowed to enter, or the antigens are allowed to enter, you can see it's kind of a trap, because right behind them is a dendritic cell waiting to just totally destroy whatever it is that gets in. Well, when that dendritic cell sucks in the antigen, it will then show it to B cells, it will show it to T cells, and in this specific instance it will make IgA antibodies, and IgA antibodies are good at neutralizing things, so they'll come and they'll go bind to the antigen everywhere in the body, and when they get bound to the antigen, it makes it so the antigen can't get into anywhere. It kind of neutralizes it. So that's another way that antibodies work. IgA antibodies, they're usually secreted into the mucus. You also find them in tears and saliva. Okay, but generally neutralization would happen, in these areas, the mucus, tears, and saliva, but can also happen in other places of the body through IgG binding. Okay, IgE are usually considered the antibodies that are involved in allergic responses. So these will be things that bind to some allergen of some kind or perhaps a parasite, and then they create a nice handle for mast cells and basophils to attach to, and then these usually are involved in eliminating, or suppressing an allergic response. The IgD is actually a B cell receptor. So when there's a B cell and it has a B cell receptor, what it is an antibody, but specifically a class IgD antibody. Occasionally, too, there can be an IgM, but specifically just the monomer of the IgM, so just one of these. So B cells could have an IgD or an IgM antibody as their B cell receptor. As a review, lots of the classes of antibodies, but their main effects are to stimulate complement or to neutralize things, to prevent them, coat them and prevent them from getting around in the body, to opsonize things, coat them and make them more easily phagocytized. And then a fourth one that I didn't quite specify but alluded to is this process known as agglutination. And this is advantageous because it takes hundreds or thousands of pathogens and clumps them into one big ball, so that the immune system only has to get rid of one big ball of bad stuff versus lots of free roaming things. That's really well done here by the IgM antibodies, because again they can go and they can just agglutinize the heck out of stuff and put them together. But agglutination can also happen with all of these other antibodies because they have more than one binding site. Okay, we're almost done here, so we're just going to finish up on a few things. One is talking about the primary versus secondary responses. A primary response, what happens is it's the first initial response, and we'll have, the antigen binding to the B cell and it activating. This is showing a T cell, T-independent B cell activation because there's no T cell involved. But this also happens in a T cell dependent B cell activation. But what happens is when the B cell is activated, it forms clones. Some of those will become memory cells, but a majority of them become plasma cells, and then they will release tons of antibodies. Okay, this, because there aren't memory cells already standing by and we have to go through this B cell activation process, this is a little bit more of a slower process. Also, what's usually released is IgM antibodies. IgM antibodies over time will class shift and usually become an IgG antibody. That happens because these are really big and they can't get into everywhere. These are really small, so they can get into everywhere. But again, these are really big. They're agglutinizing machines, so they can soak up pathogens right away. These, while not so great at agglutination, are very mobile, so they can get into different parts of the body. But in either case, that's a primary response. In a secondary response, there are already a whole bunch of these clones, the memory cells, that are on hand, and so this reaction can happen a lot faster without the whole activation process. When you look at just the sheer amount of antibodies that are released, a primary response will have so many antibodies that are released, and then it comes down. And then if there's a secondary response, because there's so many memory cells that each get activated and release antibodies, the number of antibodies is much greater because of that extra antibody production capability of those memory cells. Usually, this is what I used to learn, but maybe this is changing, and that's why they haven't included this in the figure. But usually the amount of time that it takes for the response is different, so this takes a little bit longer, and then here this happens faster, but that isn't quite reflected here in this graph. So I don't know if that's a concept that has changed as we've learned more about these responses. Another thing to talk about briefly here is the difference between what we call passive and active immunity. Passive immunity is one that is the immune response is not activated in the individual, but they receive antibodies that have been made by some other means. An active immunity is one where the individual has had an immune response and they are making their own antibodies. Examples of this, an active, and also these can either be natural or artificial. So an example of a natural active response would just be a normal adaptive immune response, anything similar to what I've just been talking about. But we can also introduce antigens on purpose into the body to create a active response, and this would be an example of what a vaccine is. In a vaccine, there's a variety of different vaccines, but they'll usually take the antigen of the disease-causing pathogen and introduce it into your body. Sometimes they've weakened it, which would be called a attenuated vaccine. Sometimes it's totally destroyed and killed it, or sometimes it's just a small fraction of the pathogen. But in either case, it will elicit an immune response artificially. There can also be passive immunity. An example of this is when antibodies cross the placenta and go into the baby that is developing in the womb, or during breastfeeding. When my wife was breastfeeding our boy, he didn't get sick at all. And as soon as he was weaned, he started getting sick all the time. So I think I attribute that partially due to the passive immunity that was being conferred by my wife. My wife has like an immune system of steel. She like never gets sick. So Wally was getting some pretty good antibodies in that breast milk. Another example though here would be like an immune globulin injection, and this is kind of like an example here that we've talked about is RhoGAM. In RhoGAM, we're giving synthetic, artificial antibodies, those which have been made in a lab of some kind, and we're injecting them into the body. So it's not a real immune response, and furthermore, it's an artificial antibody, not one made by the body. Okay, the last thing to talk about is disorders of the immune system or immune responses. And sometimes there can be hypersensitivities, meaning that the immune system is too robust, it's too vigorous. An example here is allergies. Some people have allergies to different foods, and that's a case where the immune system is attacking things that aren't necessarily bad, so the immune system is a little too hypersensitive. There's a whole bunch of different allergies out there, so we won't spend a whole lot of time talking about all the different kinds. There are also immunodeficiencies. One example here is severe combined immunodeficiency disease, or SID. And this is where T and B cells are lacking. So we're not able, the individual would not be able to mount as sufficient of immune response as they should. This one you're probably familiar with, acquired immunodeficiency syndrome, AIDS. This is a disorder where specifically the helper T cells, or the CD4 cells, are being targeted. This is usually, we talk about AIDS in combination with a certain kind of virus, which is known as the HIV virus, because HIV is going to destroy helper T cells, and then the disorder that results after that is called AIDS. Now, if there was other viruses that destroyed helper T cells, we would also call those variations of this disorder, this syndrome, acquired immunodeficiency syndrome. There are also autoimmune disorders, and autoimmune means the body attacks itself. And usually this has something to do with either antibodies that are being made because the B cells had B cell receptors that bound self-antigen. It's usually more involving the T cells. They have a more rigorous training protocol in the thymus. And so usually an autoimmune disorder is a result of a T cell which has gotten out of the thymus and binds self-antigen. But not a lot is known about autoimmune disorders, the very specific causes of it. It could be because a B cell or T cell didn't have the tolerance that it should. It could also sometimes be due to virus. And I will kind of share a personal story. I had chicken pox really late in life. When I was 26, I got chicken pox and I got a bad case of it. And those kind of viral infections that get into, nooks and crannies of your body and invade a lot of the different important cells in your body, it can then be presented on the surface of those cells, and then your body can start attacking those cells, which previously it didn't attack. So, it can get, in the case of chickenpox, it gets into your nerves and your neurons, which generally aren't infected by other viruses, like a cold virus or whatnot. So I've had kind of a lot of weird health issues. I mean, I still am grateful and lucky for the health that I have. I'm pretty functional, but if I get tired or I get some really goofy neurological symptoms, and just kind of had a smattering of other symptoms as well. And I attribute that partly because there's going to be some chickenpox virus still in my body that elicits immune responses against my own organs. There are other examples of autoimmune disorders, for example, lupus, type 1 diabetes. This is where the body, for whatever reason, starts attacking the pancreatic cells that produce insulin. And then another example is rheumatoid arthritis. This is where the body starts attacking joints and causing inflammation of joints. So those are a few examples of autoimmune disorders. Here's where we will end our discussion of the lymphatic and immune system. I hope you'll have a far greater appreciation for all that's going on right now in your body to keep you healthy. And email me if you have questions.